For this second segment, we're going to turn our attention to the brain itself. Here we have 100 billion dynamic neurons surrounded by 10 to 50 trillion neuroglial cells. And each one of the neurons has 1,000 synapse connections with other neurons. That means that we have 10 to the 15th power possible transmission sites of information within this brain. That's more than there are stars in the galaxy. And all of this is only 2.5 pounds. So how do we protect this all-important brain? First, we have the scalp. And if you look at the letters in the word scalp, you'll remember the layers of it. The S stands for the skin, C stands for the connective tissue, A stands for aponeurosis, which is the tendon sheath that connects the occipitalis with the frontalis muscles. Then we have L, and that stands for loose connective tissue. Then we have P, the pericranium, or periosteum on the actual skull bones. Then we have, of course, the layer of the bone, or the cranium, which is very protective of the brain itself. And then we have the meninges. The meninges are made of 3 layers, and they're tough, fibrous connective tissue layers that protect the brain. The first layer, or the outermost layer of the meninges, is called the dura mater, and that stands for tough mother. Underneath that, we have what we call the arachnoid matter. And arachnoid, of course, stands for spider, and this is a more loosely knit connective layer that also has a space called the subarachnoid space beneath it that is filled with cerebral spinal fluid. And that fluid is very protective, of course, of the brain as well and helps to cushion it. Underneath the subarachnoid space, we have the pia mater. And the pia mater is a very thin and more fragile layer of connective tissue that covers the actual cortex of the brain. Pia mater stands for gentle or intimate mother. Let's look at the functional regions of the brain. Starting from the bottom, we have the spinal cord shown there in yellow. It connects to the brain stem. The brain stem consists of the medulla oblongata, shown there in pink, the pons, shown in green, and that yellow region called the midbrain. The function of the medulla oblongata, the lowest region there, is centers of breathing control, heart rate. It also controls the functions of vomiting, coughing, sneezing, hiccuping, and swallowing. A very important region of the brain that's unconscious but vital to survival. The pons, just above it, the green area, has centers for modulating the breathing rhythm, as well as the control centers for many of the cranial nerves. The cranial nerves we'll talk about later on. The midbrain, just above that, is a region that has the control centers for the movements of the eye and the pupillary reflexes, and this controls the amount of light that comes into the eye. The next region, the diencephalon, shown there in purple, has 2 segments, the thalamus and the hypothalamus. The thalamus is an extremely important region for sensory perception and emotional control. It's an unconscious region of the brain that channels all of the sensory input from the body and then relays it to the higher centers in the cortex. It also helps to originate emotions. So this is why a lot of times we have an emotion perhaps before we're almost consciously aware of the sensation that's causing it. And that makes it difficult sometimes for us to consciously control our emotions. But it is, of course, possible. The hypothalamus, below that, is an important link between the nervous system and the endocrine system. It's the master gland and controls the release of most of the major hormones that control endocrine function throughout our body. It also has centers for controlling body temperature, appetite, wakefulness, and pleasure. The next region we'll look at is the blue area there. It's called the cerebellum. The cerebellum has 2 lobes, just like the cerebral hemispheres, and is connected by what we call a vermin, which looks kind of like a big worm between the 2 sides of the cerebellum. It's highly convoluted, has a large surface area. It's important for coordination, balance, and posture. Without the cerebellum, or if there's damage to the cerebellum, a person basically appears to be very drunk. Their movements are not smooth or coordinated. The last major region that we're going to look at here is the cerebrum, and this is the largest part of what makes up the human brain. The cortex of the cerebrum, or the outermost covering, is only about 1/12 of an inch thick. but it has 6 layers of cell bodies and dendrites where most of the conscious processing of information that occurs in our brain occurs. It has a very wrinkled surface and is made up of 10 different lobes that are divided up by different fissures. We'll look at those in the next slide. The cerebrum is divided into 5 lobes, 5 on the left, 5 on the right. These lobes are divided by large fissures, or indentations, and then each of the lobes are divided further by little creases called sulci, and the bumps that you see coming up are the gyri. Various regions of the brain are mapped and numbered according to their primary functions. The overall function of the cerebrum, of course, is conscious awareness of sensations, and then response to those sensations through voluntary motor control, memory, learning, and communication. Now we'll move down to the spinal cord. The spinal cord includes a lot of gray matter, which is in the internal part of the cord, and the outer part of the cord is the white matter. So cell bodies are present in the spinal cord. It's not just a group of axons. The outer part, the white part, is made up of tracts that conduct information from the central nervous system down and then out to the peripheral nervous system. There are 31 pairs of nerves that leave the spinal cord, and these help to form the peripheral nervous system in conjunction with the 12 cranial nerves. 8 of the nerves exit from the cervical region of the spine, 12 from the thoracic region, 5 from the lumbar, 5 from the sacral, and 1 coccygeal nerve. Nerves exiting the spinal cord at the cervical region, there's again 8 of them, form networks called the cervical and the brachial plexus. Basically, a plexus is much like a freeway interchange, with some of the nerves merging and others diverging, and mixes the axons coming from the various spinal levels and sends them on as peripheral nerves to the sensory organs and muscles. The lumbar and sacral nerves also form plexuses, but the thoracic nerves do not. They just innervate the organs and muscles at the same level that they exit the spine. Although nerves belonging to the sympathetic nervous system form a ganglion chain parallel to the spinal cord on both sides of the thoracic spine. We'll talk more about that aspect when we talk about the autonomic nervous system. The spinal cord is also covered by meninges, the dura mater, the arachnoid, and the pia mater. The spinal cord itself ends around the level of L1, but below that the spinal nerves come out and go down to their lower levels where they're going to be actually exiting and form something that looks like a big tail on a horse, and that's what its name is, the cauda equina. This short segment of the thoracic spinal cord shows the nerves exiting at the various levels, as well as the formation of the autonomic nervous system's sympathetic chain ganglion, which we'll hopefully have a lot of time to talk about in class. Now we'll talk about the ventricles. These are open spaces within the brain that are filled with fluid. There's 4 ventricles, 2 lateral ventricles that you can see well in this picture. They have an anterior, posterior, and an inferior horn. They're connected in turn to the 3rd ventricle, which is between the right and left sides of the thalamus. Then the cerebral aqueduct drains the 3rd ventricle into the 4th ventricle. The 4th ventricle is kind of a diamond-shaped region just anterior to the cerebellum. From the 4th ventricle, there are spaces that open up into the subarachnoid space, and the fluid can then circulate around the brain. There's also a central canal that goes down through the spinal cord. Here we can see where the fluid is produced, in the choroid plexus of the 3rd and lateral ventricles. Here, cerebrospinal fluid is secreted by specialized cells and fill the ventricles, the lateral ventricles, the 3rd ventricle, draining into the 4th ventricle, and then out into the subarachnoid space. And you can see how that circulates that way. From the subarachnoid space, in the upper part here of the cranium, you can see how it is reabsorbed into the venous sinuses surrounding the brain. The venous sinuses are actually between the layers of the dura mater. Specialized, what we call choroid villi, are placed around this area, and they reabsorb the cerebrospinal fluid back into circulation. So you have a constant circulation of fresh cerebrospinal fluid being created in the ventricles, circulating around the brain, and then being reabsorbed to the choroid villi. If anything blocks this process, for example a blockage in the cerebral aqueduct, it causes swelling inside the brain. And in very small children where they have unfused cranial bones, this can cause a very enlarged head, and we call that hydrocephalus. This is why measurements are usually taken of a newborn's head many times to see that it's not swelling.